Need to share my slide. PowerPoint. Okay, so everybody can see my slide, right? Engkau? Yeah, okay. Okay, so for my part, that would be, I think this part is questions 3, if I'm not mistaken, question 3 or question 4. It would be have two questions from my part and then 2 questions from Dr. Shohayu. So today's we are going to highlight on what on the important part lah, so that you can answer the exam questions very well. Okay. Okay, so the topics for today is DNA sequencing and application in molecular medicines. Okay. So basically this topic at the end of the lecture, you should able to describe the example of the DNA sequence techniques, and then you should be able to describe the process of DNA sequence analysis, and then explain the applications of DNA sequencing in molecular medicines. Okay, so what is sequencing? Okay, so you have to know what is sequencing. And before that, I think both of you already know that we have DNA and also RNA, right? So if you see here, this is the DNA. It is the double helix strand of DNA, and RNA is basically a single strand, right? So what are the difference between DNA and RNA? Anyone? Gao, all right? Can you tell me what the difference between these two?
The base.
Hello.
Yeah, hello, I can hear you, yeah?
Yeah.
The DNA and RNA difference between the sequence and the nucleotide.
Nucleotide, right? So the nucleotide is, what are the nucleotide that difference? In RNA, we have uracil, right?
Yeah.
So you can see here in RNA,
Thymine.
yes, DNA has thymine, and DNA is double strand, right? But RNA is a single strand. Okay, so because we have these informations, the nucleotide informations for DNA and RNA, so that's where the sequencing technologies develop. Okay. All right. So what is the sequencing? It is the process in determining or identify the order or the arrangement of these 4 nucleotides. What are the 4? There are adenine, guanine, cytosine, and thymine, which which make up the DNA molecule. Okay, so this the sequence contains the genetic information that is carried in particular DNA segment. So you clear about that, right? You understand what is sequencing, right? So actually it is a process to identify these 4 nucleotides in the correct order. Okay? Okay, so why we need to do DNA sequencing? Okay, the first one is to do comparative genomics. With the comparative genomics, you will learn about the evolutionary history of different organisms, and we need DNA sequence to identify the genetic that causes any disease, cancer disease, infectious disease. So we will use DNA sequence to determine the genetic, what are the mutations or single nucleotide polymorphisms needs that causes the disease. And DNA sequencing also used to identify a potential drug targets. In forensic science, we use DNA to identify criminals and paternity of children. Also, we do the DNA sequencing for functional genomics studies and also metagenomics. Okay. Okay, so this is the NGS sequencer technologies. Okay, so before we have NGS, NGS stands for what? Next, do you know what NGS is? NGS, right? Raul, Gao?
Next generation sequencing.
Yes, correct. So what are the next generation sequencing platform? It is second generation and third generation. The first generation is not considered as NGS. Okay. All right. So we will look at more details on each of these generations. And you need to know the difference between these three generations. What are the difference between the first, second, and third generations? Okay. So NGS platform can be largely divided into two bases of the length of the sequencing output. Okay, so for NGS, we have short reads and we also have long reads. For short reads, meaning that it will sequence in very short fragments. Remember last lecture I talked about Illumina. So example of the short read platform is Illumina. And another example, MGISIC and Ion Torrent. Okay. When I mention short reads, meaning it sequence only short fragments, less than 1,000 nucleotide sequence. Okay, less than 1,000 nucleotide bases. All right. So that is the meaning of the short reads. If a long reads, it can go up to 10 kilo bases of nucleotide. All right. So far so good. So the example is PacBio and Oxford Nanopore. Okay, so this is the first generation sequencing technology, and the example is the Sanger sequencing. It also known as a dideoxy chain termination sequencing. Why it known as a chain termination sequencing? Because it use dideoxy DDNTPs to terminate the DNA synthesis. Okay. And all Sanger sequencing it used 4 separate reactions in different tubes. It will have 4 different tubes. So each of these tubes will contain all 4 DNTPs and a single DDNTP. But the current technology of Sanger, we use microfluidic technologies and it uses fluorescently labeled DDNTP, which allows Sanger sequencing to be separated through capillary array electrophoresis, and it can has achieved a greater throughput. Okay, so this is just to show you how the Sanger sequencing, the mechanism of the Sanger sequencing. So basically in Sanger, you need to have a primer, prime to the 3' and then you need the single strand of DNA template. Okay, so this is basically your interest DNA. And here is the DNTP. So we need DNTP so that the DNA can be synthesized further. Okay, and then in Sanger, we have DDNTP. So what are the functions of DDNTP? Gao, what is the functions of DDNTP?
Primary?
Sorry?
Primer?
Primer?
Yeah.
No, this is primer. Primer, we need primer so that it will attach to the DNA template. So this is the one that we try to sequence. DNA template is our interest DNA that we try to sequence. Okay, so we need a primer to attach to this DNA. Okay, so when the primer attach DNA to the DNA, so the nucleotide, the newly nucleotide here will be synthesis. And DDNTP is actually to terminate the DNA synthesis, so that the DNA will stop to produce. If you see here, this is primer, and then it will synthesize the nucleotide based on the template. DNA polymerase, okay, will synthesize the newly nucleotide using the template. Because remember that I told you Sanger sequencing is a chain termination sequencing, right? It terminates the DNA synthesis. Okay, so DDNTP will incorporate the newly synthesis sequence, and then it will terminate at the what nucleotide position it have. For example, if this position is A, so the DDATP will incorporate here, and it will terminate at A nucleotide. Okay?
So just as a primer.
Yes, you need a primer. You need the primer to start the DNA synthesis. Okay? And then DNA polymerase will incorporate it here. It will synthesize the newly nucleotide. So how do the DNA synthesis stop? How do the process of extensions of the nucleotide stop? Is basically using DDNTP. So DDNTP, it will have DDATP, DDCTP, GTP, and DDPTP. So it basically will have 4 deoxynucleotide lah. So because why we need 4 NTP is that because if DDATP, it will stop the reaction at A nucleotide. If DDTTP, it will stop the DNA synthesis at T nucleotide. So far, so good? Or are you still confused?
That's
Okay. Good.
Okay. Okay. If you have any question, just you can just interrupt me. No worries. All right. So another example of the second generation's generation sequence technology is a Masson and Gilbert. So this technique use very dangerous chemical. So it terminate. The use of this method have been terminated because of the dangerous chemical being used. And the cost for sequencing have been decreased dramatically every year by year. If you can see, that started 2008. So the cost is very has been decreased. So this is because we have the next generation sequencing platform that have been developed. So next generation sequencing is remember it is second and also third generation DNA sequencing. Okay. Okay, so what is next generation sequencing? It is a modern high throughput DNA sequencing technologies. It can sequence the DNA or RNA in parallel fashions, and it can sequence rapidly. Because of the characteristic, right? It can sequence many samples in parallel fashions, and then the process more quicker, so it will decrease the time, it will decrease the price, it will decrease the workflow complexity and also the error rates. And the good thing is that NGS will increase the data quantity and quality of the read. It also increased the read length. And when it increased the read length, meaning that you will have many data, a lot of sequencing data. So can you analyze it manually? It's almost impossible. It's not impossible. I think it's not impossible for you to analyze it manually using your own eyes. So that's why we need the bioinformatic tools to do the sequence analysis. Okay. And what are the difference between the first and also NGS? Right? What do you think? What are the difference between first generations and NGS?
What mean the first generation use the difference between the DNA, RNA, and primary and application they use the.
Okay, the first generation, you cannot using many samples to sequence at one time. You just only can sequence one sample in one time. But with NGS, you can sequence many samples in one time.
Okay, that's all.
So that's why this is what I mean when I said parallel. It can sequence many samples at one time. Okay, so when you sequence many samples at one time, it will produce many data, right? Many sequence data. So you will have a big data for that sequence. But with first generation, you just only sequence one sample. So the number of the sample is less compared to the NGS. Okay, NGS, you will have many samples. You can sequence many samples. But first generation, you can just only sequence one sample. Less sample compared to many samples with NGS. Okay, all right. And the time to sequence with the first generation, it also takes time. Okay, but with NGS, it can sequence very quick. It happens in rapid. And NGS has a wide range of applications. It can be used in gene expression studies, where you can identify the rare transcript without prior knowledge of a particular gene alternative splicing identifications. It also can be used in large-scale comparative and evolutionary studies. And also we can use NGS to resequence human genomes. Okay, and in next generations, we have now third generation sequencing, where we use single molecule and it happen in real time. So what do you think the meaning of the single molecule here? Single molecule.
First generation.
Yes, it's not generations, but we use only single strand DNA. Compared to the second generation, you will need to amplify the single DNA into many numbers. But with third generation, you just use that single strand DNA. You don't need to amplify to many copies. But with second generation, you need to amplify it so that it can become many sequence template. Do you understand what I mean? Single molecule mean, remember that we have a single strand DNA. We need to have a single strand DNA. The originally DNA is double strand, right? If you want to do sequencing, you need to break the double strand into the single strand DNA. So you will use only single strand DNA for sequencing. Okay. For third generation, you use that one single molecule, one single strand of DNA for the sequencing. But for the second generation, like Illumina, you need to do the amplifications of that single strand DNA. So it will become many copies of DNA. Okay? Is that okay?
Yes.
All right. So the NGS workflow, of course, you need to isolate to do the DNA isolations. Okay. For example, if you have the bacteria, so you need to isolate the DNA of that bacteria using methods lah, using experimental methods. Okay. And then you need to prepare your DNA with library preparation. So this library preparation will be different. If will be different based on the sequencing platform that you want to use. And then you send it for sequencing. If you want to use Sanger, so you for the library preparations, what I mean different platform, different library preparation is that if you use Sanger, so it will have a certain or particular chemicals that you use for library preparation. It will different with the library preparation that from the Illumina platform. Okay, so and then after that, after sequencing, you need to do the data analysis. All right, so now we come to the third generation sequencing technology. So I will not talk about second generations more details because today I just want to highlight the first generations and also the third generation sequencing technologies. Okay, third generation sequencing technology is actually similar to the Illumina, but it doesn't involve any bridge amplifications. Okay, when I say about bridge amplifications, meaning that it will not make the sample or the DNA, single strand DNA, into many copies. It will not amplify it into many copies of DNA. It uses only single molecule, one molecule of DNA, one strand, one DNA strand for sequencing. And of course, the price is more expensive compared to Illumina and also first generation sequencing technology. Okay, so here Sorry, the first example here is a PacBio. So PacBio is one of the third generation sequencing, one of the example of third generation sequencing technology. So for PacBio, it uses a single molecule of DNA, and it happened in real time. Okay. So here, if you see here, this picture is actually the SMART cell. The SMART cell used in a PacBio to load your sample. So here, you can load your sample in the SMART cells. So what happened in the SMART cell is that, if you look at this figure, at the bottom of the SMART cell, it contains DNA polymerase. This is the DNA polymerase. It's static at the bottom of the SMART cell. It cannot move. It has been immobilized at the bottom of the SMART cell. So when you have a DNA template, so it will, the single strand, the double strand here will break, and it will become the single strand. So the single strand, and then DNA polymerase will synthesize the nucleotide based on the single strand of the DNA template. Okay, and also PacBio, we can use PacBio to identify the methylations. So the how it detects the methylation is that based on the duration time of the detections. If the longer detection time for that nucleotide, meaning it has the methylations. And then another example of third generation sequencing is Oxford Nanopore. So it is the same as the PacBio. It uses only a single molecule for sequencing. So this is DNA double helix. So it has a one protein here. So the functions of this protein, the first protein in nanopore, is to unzip the DNA helix into two strands. So only one strand will be used for the sequencing process. Okay. So this is the example of the Oxford Nanopore technology. We have launched MinION, GridION, PermION, and MinION. Okay, so the advantage of using Nanopore, you just only require a simple sample preparations. If you see at this slide, okay, it's only take about 10 minutes to prepare your sample before you sequence. Right? Okay, and it produces very long read lengths, more than 10,000 to 50,000 nucleotide. And the instrument is inexpensive compared to the PacBio. Very portable, meaning it can take, you can, because it's small in size, it's like your pen drive. So you can take anywhere you would like to take it, and you can work whether in your lab, or you can take it to your room, any place, because it's not big. It's came in a small size, and it can produce very high depth and coverage. So this is the challenge with the nanopore, is that to slow down the DNA translocation from microsecond per base to milliseconds. Okay, so after this lecture, so what do you think of the different characteristics each generation of sequencing technologies? So we have first generations, just forget about second generations and focus on third generations. So what do you think the difference between the first and also third generation sequencing technologies? Any idea, right?
Doctor, I think the difference between the for the generation about work, yeah, the specific, the make the something, the technology.
Anything you think of that is the difference between the first and third generation. Anything you can just think of anything after you listen to the lecture just now. What do you think the first generation? Do you remember the first generation DNA sequencing? The example of the first generation DNA sequencing? It is? What
think
are the example of first generation sequencing? Sanger. Did you remember Sanger? No, not remember. I just give the lecture just
什么是数量？
Sanger. So the example of first generation Sanger. So Sanger, it produce short or longer reads. Short reads, right? Do you agree with me? It produces short reads, right? Short, less than 1,000 nucleotides, right?
Yeah.
Okay. Okay, what about third generation? The example of third generations. Can you name the example of third generations? PacBio and Nanopore. So in terms of the reads length, it produce first generation, it produce short reads. Third generation, it will the opposite to the first generation. It produce longer reads. Right? Do you agree with me?
Longer what? Waves?
Longer reads, longer sequence.
Or reads? Oh.
Yes, we call it reads or longer sequence, a longer nucleotide sequence. You can say, or otherwise, you can say as a longer reads, produce longer reads. First generation, shorter reads. Third generation, it will produce longer reads. And the first generation will take time to produce the reads, the sequencing result. But third generation, it can sequence very faster, more faster compared to the first generations because of the advanced technology that we have now. Okay, and first generation, you cannot sequence many samples at one time. Okay, but third generation, you can sequence many samples at one time. Right? So that are the obvious characteristic between these two lah, right? Okay, so this is here. The first generation is actually the start of sequencing technology. Low throughput. Low throughput meaning that it produce smaller data size. Low throughput means it sequence smaller samples. Okay. Second generations massively multiparallel requires amplifications, which I already mentioned about Illumina. And third generation single molecule sequencing, and it happened in real time. Okay, so this is the difference between the first and third generation technologies. So in terms of read length, okay, for first generations, it can be used for a smaller sample size. So it can be used to target sequencing project or for its high accuracy in sequencing individual genes. Only one genes, not the whole genome. Okay. So if you have only one genes, do you understand genes and genomes, right? You know the difference between genes and genome, right?
Right.
Right. Okay. For the first, in terms of read length, for the first generations, it can be used to target only certain genes, in order to get the high accuracy result. You cannot use that for the genome study. If you want to do the genomic study, the whole genome study, so you need to choose the third generation's technology. So you can use PacBio or Nanopore so that you will have the complete whole genome for your organism, right? In terms of accuracy, okay, for the first generations, for the normally in clinical diagnostic or targeted gene sequencing, right? So the accuracy is very important. So you can use the first generation's sequencing technology like Sanger. Okay. But for the third generations, you need to sequence many times in order to improve the accuracy. Okay, let's say what I mean many times. Okay, one sample, so you need to sequence that many times. So using third generations, it will sequence that one sample many times, so that when you do the analysis, you will have like millions reads, the similar million reads, and you overlap each other to improve the accuracy of the reads. Okay. So if your studies want to have high accuracy, so that you need to use the first generations. But saying that, that's only focus on gene, not genome. Because first generation, you cannot use it to sequence the whole genome of that organism. You only need to target certain individual genes for the first generations. And if you are using that first generation for the gene, it will give higher accuracy, right? Okay, is that okay?
Yeah.
So what about the cost? In terms of the cost, what do you think the cost for the first generations? Expensive or cheaper?
I think cheaper because use a
Yes.
small and
Yes, correct, right. Yes. The cost for the first generation, of course, it's become more cheaper lah, because it use smaller,
Yeah.
smaller sample size, and it use only targeted studies.
Sequences.
Targeted studies, you target only certain genes for sequencing, right? Compared to the whole genome project, for large scale project, so you need more money to sequence or to do that. Okay. Right. What about throughput and speed? Throughput and speed for first generations. So for the first generation, yes, right. You want
lose
to answer
time
that?
sensitivity objective. I think sometimes not correct because it's speed.
Here means it's not about the accuracy. Sorry. Actually, the accuracy of the first generations is more accurate because you just target only one gene compared to many genes in one genome, right? So the accuracy is more accurate. Less time sensitive project, meaning that it doesn't require immediate attention or has visible deadlines. So if your project, if your project is not really important, you don't need to know the result very you don't need to know the result in a faster time. So you can use first generations, okay? But if you have something that very urgent, something that very urgent, like here, for example, for pathogen surveillance or urgent genomic analysis to know what are the disease having causes to the mortality. When the patient have been infected with some disease, and then for the outbreak something like outbreak of infectious disease, so you would need the third generations to do the sequencing so that you will know what are the bacteria that causes the disease. Okay.
Yeah.
All right. Okay, so mechanism of PacBio and Oxford Nanopore. So this is the mechanism of PacBio and Oxford Nanopore. For PacBio, it uses zero mode wavelength to detect DNA synthesis in real time. Compare Oxford Nanopore, it's used nanopores. Remember the proteins, the two proteins, the first protein and the second proteins? Okay, so it used the nanopores which are embedded in a membrane. Okay, it's different with the PacBio. And in a PacBio, DNA polymerase incorporates nucleotide and fluorescently labeled phosphates. It will be cleaved off and then it will generate a light. So that is how the nucleotide bases have been identified in PacBio. Okay.
Yeah.
Okay, can you understand that?
Yeah.
Okay, here. Okay, here. All right, here. When the nucleotide have been created here, the light will be fluorescence. And it has a camera at the bottom here, and then it can detect the nucleotides. What are the type of nucleotides? Whether it's G, whether it's A. And if you see, if G, it has been labeled as a blue color, different color. So each of the four nucleotides will be labeled as a different color. Okay. And for Oxford Nanopore, the DNA or RNA will pass through the nanopore, will pass through the protein, and the changes in ionic current will be measured. Okay. It's different, right, between PacBio. PacBio, it uses fluorescent light, so it can detect the nucleotide, whereas Oxford Nanopore, it uses ionic current to detect or to differentiate between the four nucleotides that we have. Okay. For PacBio, it will allow observations of DNA synthesis on a single molecule. So, whereas Oxford Nanopore, it will identify the sequences basis through ionic current. All right. Okay, in terms of accuracy between PacBio and Oxford Nanopore. Okay, for PacBio, initially it has a lower accuracy per read because that due to the random errors. For Oxford Nanopore, higher error rates in single reads due to the difficulty of identifying identical bases. So, combining several reads, consensus sequencing result in high consensus accuracy. So, how PacBio overcome the lower accuracy is by combining several reads that result in high consensus accuracy. Okay, remember that single molecule, it will sequence multiple times. The same molecule will be sequenced multiple times. Okay, it will have many, and then after that it will have many reads. So from there, from overlapping each reads, you will overcome the lower accuracy. Okay, so for Oxford Nanopore, it will overcome the higher error rates by improvements over time through software updates and more precise base calling algorithms lah. So it's developed more precise software for the base calling algorithms. Okay, so this is the example of the NGS applications in molecular medicine. So currently we have whole genome sequencing, RNA sequencing, dual RNA, shotgun, targeted, and exome sequencing. You don't need to know more details on that. Just for your knowledge. So WGS whole genome sequencing used for surveillance of foodborne infections. So using the whole genome sequencing informations, you can do serotyping, virulence vector, AMR dissections, and molecular typings using informations on the sequence predictions. So this is another study. It used the whole genome sequencing to determine the bacteria species and also to identify the MLST, multi-locus sequence type, using the whole genome sequence informations. And with the whole genome sequence, you can use to generate a phylogenetic, so that using the phylogenetic, you will know what are the relationships, and also you can detect the virulent genes via the software, via the tools called Virulent Finder. Okay, so I think that's all from me. Okay, so any questions, right, Gao? Do you have any question?
Victor, because don't have time, I think found 6 days for the exam. You can focus the important lectures.
Okay, so basically the important is that you need to know the first generation sequencing technology. Okay, you need to understand the third generation sequencing.
The version between?
Sorry?
The version between the first and second generation is important?
Yes, these two is important, first and third generation. So you don't need to study for the second generations.
Okay, only one or three?
Yes, only 1 or 3, and then you need to know the difference between the first and third generation sequencing technology. Okay, and then for the third generations, remember it. I give you two example, right? PacBio and Oxford Nanopore. So you need to know the difference between these two method, right?
Mm-hmm.
Okay, so that's all lah for your exam. Okay.
Thank you, Doctor.
All right. Go. Any questions from you?
No. Just exam. Okay.
Okay. All right. I think I can pass the next sessions to Dr. Shuhaila. We stop my sharing. Dr. Shu, are you there?
Yes, I am here. Assalamualaikum and good morning.
Good morning, Doctor.
Is it my voice clear?
Before that, sorry. Do you need this slide? Go and write. Do you need this slide? You want me to share with you?
Yeah, you can share with the on WhatsApp or the email.
Email. Email. Okay. All right.
Okay,
Okay.
Doctor.
Okay,
Okay,
Dr.
thank
Shu,
you.
you can continue.
Okay. Oops. Macam mana nak besarkan ni? Okay, so basically today we just do some revisions. So So I think you guys are familiar with the DNA and RNA now. Go and right, is it?
Yeah.
Oh, okay. So this is just the overview how the genome size could be different from the virus until the higher eukaryote. Okay, so the smallest genome could have the small genome size, but it's not necessarily small. Okay, because some of the organism like plant could have a more size compared to the human, for example. So we know that some of the genus, like some of the genomes have a highest genome size, not exactly by comparables with their genome size. Okay, the organism size is it? Okay, so this is just examples. Okay, the virus could have the RNA or single-stranded RNA. And basically, the overview of the genome analysis, you know that you can have the raw data from the SRA. Okay, you get the data from SRA. Okay, the SRA data sets, and then you need to do some trimming, okay, and then you get the good quality, and then you do the assembles. This is all has been the part of the analysis that you need to do to get the chromosomes or the whole scaffolds. Okay, so from these scaffolds you will do the protein predictions and annotations, because we want to see the genomes inside the organisms. What does the genes that can contribute for each of the function inside of the genomes, inside of the organism itself. Okay. Okay, so basically for the introduction, you will have to have the QC. Okay, you need to do some QC for the raw reads, and then you can do the genome assembly. So for the sequencing, I think this one have already covered by Dr. Nick Yus. Okay, so basically normally we have the raw reads, okay, that's come out from the Illumina sequencings. Okay, so the FASTQ that we get is basically that will have this kind of format. Okay, it start with the alias. Okay, HWI, this is the ID. Okay, this is the sequence. Okay, and then this is plus with the quality base. So below here This is basically the quality base. Okay, so the quality base will be recognized by the systems. Okay, for examples, alias could be 1, the question mark here could be 5 or 7. Okay, they will have their own numbering or scores. Okay, that has been assigned to each of these symbols. Okay, and normally when we do the QC for the raw rates, okay, so we need to calculate, and we need to have like a cutoff, okay, at least 20, more than 20% for the quality rate, okay, like 99% for the base accuracy. So if we have the cutoff like more than 20%, we can use that raw rates as our data sets. Okay, so this is how we can see when we do the FASTQ, like the quality check, okay, for the raw rates. So this is the good results when you can see all the data set is actually above in the green area. But if you see that all the rates is mostly in the red area, so this means that this raw rate is not a good sequence. Okay. So this is all the features that you can see, okay, once you run the FastQC, okay, and then you do the once you get the clean reads, okay, the clean reads, and then you can do the genome assembles. So genome assembles is basically you need to do the analogy here. You have the DNA from the genome itself, okay, and then you need to chop it, okay, cut into the short reads, and then you need to assemble again into the scaffolds or contigs. By then you will know the functions or the everything's inside the genomes. Okay, so the process for genome assembly will have two, okay, the de novo and also align to the reference. Okay, for the reads and contigs, okay, so basically it will start with the reads, okay, and then you need to assemble to get the contigs, and if all of this contig can be combined, okay, by adding the pair and reads, so we can have a one full scaffolds. Okay, for these scaffolds, it could be one chromosome or it could be just a long contig. Okay, so okay, so basically this one is how we do the reference assembly. Okay, we need to have a reference, which means that's the genome sequence. For example, if we have a known organism like human, we already have the reference that we can refer to. So this is much easier, and you can just use the reference, and then the reads that you get from the Illumina, for examples, and then you can do the alignments. Okay, so for the de novo, you don't have the reference. So basically, you will need to have like all the information regarding to the reads itself. Okay, you need to get all the sequence reads, and then you can just rearrange. Okay. So how we can measure whether the assembles is good or not? So you can apply the formula N50 here. Okay, so basically when we have the calculation is more than 50% of the total length, so that's mean this assembly quality is good. Okay. So for example here, if you want to calculate, for the example, we have 5 contig here. Okay, the first is 30 base pair, second is 20 base pair, third is 20 base pair, and then you calculate, you have this one is like a 19 base pair. So 50% of this base pair is actually 45 base pair. So when you calculate 30, and then the second is 30 plus 20, so it's basically more than 45. So you will have the N50 is 20 base pair. Okay. Okay, this is basically you want to compare the genome size with the genome's NG genome. Okay, NG50 is actually they compare with the genome size. Okay, so basically if the N50 is actually from the assembles, and then when we count for the N, G50 is basically the size should be include or larger than 50% of the genomes. Okay, so when we get this number of the N50, NG50 is better for the assembly quality. Okay. Sorry. Okay, so basically the N50 is basically we want to see the quality of the assembles results that you get. Okay, for depth of the coverage, okay, this the one that we need to calculate. Okay, how does we know that whether the sequence or the reads that we get, and then we do the whether that's the reads that we get, and then we assembles and we get enough read that we can do the assembly. Okay, so when you can just use this formula. Okay, the coverage is equals to the number of the reads times read length divide by the genome size. So you just need to fill in all the informations. Okay, let's say you have the 10 times reads and with the 100th base pair, and then we divide with the 10 times to the power of 6, and then you will get the 100% coverage. Okay. Okay, so after you get the good assembles or the quantity or the chromosomes, okay, so we need to do the gene predictions and annotations. Sorry. So from here, this is the examples, the scaffolds or the quantity that you get. Okay, it starts with the ATG. Normally this is the raw sequence. Okay, so if you want to identify which genes that have a function in these scaffolds, how you going to do that? Okay, do you have any idea how this sequence have genes or not? Still remember this analogy?
Yeah, you can check the sequence adenine, thymine, guanine.
Yes. Okay.
This is start for the sequencing.
Yeah. This is the result from the sequencing. Okay.
Yeah.
So from here, we do not know, right? Which of the genes is basic? Which one of the region that code for the genes, for examples. So what is the basics information that you know when you want to identify the genes? It need to have a start and stop codon. Still remember?
Yeah, you can check the number of the stop the codon.
Yeah.
Make the minus with the start codon. You can.
Okay,
You can
so
make
basically
the
you're
result
going to predict the genes, right?
Yeah.
You need to predict the genes. Okay, so that's why we get from this nonsense sequence, we can have the sense, which means in this scaffold, for example, you will have a non-coding RNA. It could be code for the genes where it start with the start codon, ATG, okay? And then TGA is the stop codon, okay? And in between of these genes, we call it intergenic regions. Okay, intergenic regions. And then you can also found the repeats. Okay, so repeat is basically the repetitive
mutation?
nucleotides. Come again?
Repeat the mutation. I see.
Yes, this one is prediction, gene predictions. And then it goes to the annotation. So when you do the annotations, you know that this is actually referred to the repeats. And this one is actually referred to the intergenic regions. And you can also found the small ORF. So small ORF is basically the small proteins that has been coded. It's similar as a gene. Gene is more than hundreds, but small protein is less than hundreds. But both of these, small ORF and also genes, is basically a gene itself. Okay, or some of these genes can be translated into a protein. Okay. Okay, any question so far? Okay, so this is the most important or the criteria or the features that you can get from the genome sequencing. Okay, once you get the chromosome or the scaffolds, you will have to do some gene predictions. So when you do gene predictions, you will get the number of the genes, number of the small ORF, or you can identify the repeats, mutations, or non-coding RNA, and many more. Okay, let's see one by one. So basically, the genes in the virus is more complex, okay, compared to the prokaryotes and eukaryotes. Okay, but most of the genes that's inside in the virus have code for the protein regions. Okay. So gene is basically, okay, genes, okay, is consists of the DNA. So DNA, we know that ATGC, right? So this DNA is actually a basic unit of the heredity, and this gene can be translated into a protein. So that's why this gene is very unique, okay, in each of the organism. Because for examples, you know that sometimes we have the brown color of hair. Okay, sometimes people have the black color of hair. So all of this is coded by the genes. So each of these genes is actually unique. Okay, it represent a unique features for each of the organism. Okay, so basically you will see that this gene is actually has been coded part of the regions in the chromosome itself. Okay. So small protein or small ORF. Small protein is basically same like the genes, but it's contain less than 100 amino acids. Okay, so you can see that most of these small protein is basically can be located in the intergenic regions, in the upstream, in the downstream, or it could be in the overlapping, but different ORF. So basically, when you do a translation, you already learned, right, how to do the gene translations. You still remember? You still remember how to do the gene translation? From the gene itself, you need to translate into the seed frame of the protein. So this seed frame, we call it open reading frames. There will be 3 forwards, 3 reverse. You still remember? You still remember? Not in my lecture, but in the in the gene prediction lecture.
I think, yeah.
You still remember?
Yeah.
Okay, so that's why it we get this information where we can have the small protein is actually has been overlapped with the existing genes, but in different open reading frame. So it's basically could be this gene is in the 3' prime, but this ORF, small ORF, could be in the 3rd reading frame. Okay, so this open reading frame here is basically the one that has been translated from the genes. Okay, these genes will be translated into the 3 proteins. Okay, it will be translated into the 3 forwards and also 3 reverse. Okay, okay. So this small protein is basically have a very good function. Okay, in terms of the regulatory and also biosynthesis. Okay. So the repeated, okay, repeat is basically, we call it a pattern. Okay, when you see the number of the sequence is actually repetitively happen in the genome. Okay, so repeats is basically a pattern of DNA that repetitively occur in the genome. Okay, so when we want to identify the repeats, okay, so this repeat is basically the one that have occur in the telomeres, in the terminals repeats. Okay, so we know that's a most, yes. or
Okay, Doctor.
or it could be happen in the short tandem repeats. Okay, so this is the regions that most of the researcher has been identified to see whether this organism have satellites, mini satellites. This is a like we call it like the uniqueness or the uniqueness of the organism itself. Okay. Repeats, we have previously small proteins, repeats, genes, and also we can have the mutation. Okay, mutation is basically a change in the DNA sequence of an organism. So the mutation can be a result from the error in the DNA replications. Okay, it could be happen during a cell divisions, or it could be happen because the exposure to the mutagens or by a viral infection. Okay, so normally like when for example the SARS-CoV-2, okay, it's basically have a highly mutations. Okay, so for examples in here, okay, it have the genome itself have a highly mutation sites, okay, that can increase the affinity of this virus to infect the cells. So this is how this virus can survive in the host or in the human genome. Okay, so
Doctor, you can the COVID make
yes.
the mutation after the
Yeah.
infection?
Yes.
Yeah.
So yes. So this virus can change the or can modify the genomes or the
Yeah.
genes in the human body.
Yeah.
Yes. So because it have a highly mutated, so it can use the human host as their reproductive system. So that's why every part in the human body, for example lung, okay, have a lot of these the dead cells that because of this replication of this virus. Okay.
Mm.
Okay.
Okay, Doctor.
Okay, but this is the example on the virus. But basically, the mutation is because we want to see the change of the DNA. So if you want to see, for example, human genomes, okay, human genome also have mutations where it could lead to like autism disease, okay, or autisms or the syndrome Down, okay, that could be happen because of the change in DNA in the human, for examples. Okay. Okay, next is non-coding RNA. So non-coding RNA is basically the one that can regulate the helps in the regulations in the genome. So it could have like transfer RNA, like tRNA. Okay, so this tRNA basically will brings the ribosome, the amino acids to the ribosome because it want to create a protein molecules. Okay, the second one could be the ribosomal RNA, or in short, we call it rRNA. Okay, so this is the function same like the protein synthesis. Okay, but you can also find many more of the non-coding RNA in the genome. For examples, this one. SNO RNA, okay, long non-coding RNA, okay, circular RNA, micro RNA, mRNA. Okay, this mRNA is basically the gene itself, but has been translated into a protein. Okay, okay. Okay, so you can also find many more of the gene predictions. Okay, it could be come from a transgene. You can identify the intron. Where you can get the intron region? Where do you think you can get the intron region? Do you know the big difference between the prokaryote and eukaryote?
Yeah.
What is the biggest difference between the prokaryote and eukaryote in the genome's sequence?
Mm.
Tak ada pula contoh. Oh, this one. Okay. So basically, in the eukaryote, we can have the intron. Okay. In between the genes. But if you have the genes or you want to predict the gene from the virus and prokaryote, you wouldn't have the intron. Okay. Clear?
Yeah.
Okay. So if you want to predict a genes, okay, from a genome itself, so you need to have the basic understanding, like it need to start with the start codon and then stop with the stop codon, okay, and then it need to have have five prime or 5' to 3', or it could be start from the 3' to the 5'. Okay. So this is the examples of how does the prokaryotic genes, okay, it doesn't have the intron. Okay, but in the eukaryote, it have the exon and intron. But during the translation, it need to remove the intron side. Okay, they need to remove this one. Okay, and only intron will be assembled in here to be a gene for the eukaryotic genes. Okay, so there will be many methods that you can use to predict the gene predictions. Okay, so basically you can have three different methods. The first is we call it ab initio or intrinsic. So if ab initio, which means it doesn't have the information. Okay, it doesn't have information. So if ab initio here, which means that it only relies on the information or what we call a standard information that we assign. For example, have a promoter information, start and stop codon, okay, the transcription binding, and also the possibles of the intron splice sites. Okay, so the ab initio is basically, it depends on the signal within the DNA sequence. Okay, this signal is actually you can refer from here, where you need to assign the because all of this feature is actually standard and like a same pattern. Okay, for example, the promoter, they have their own sequence that can be assigned as a promoter. Start and stop codon, basically start will start with the ATG, but some of the prokaryote, it can also start with the AGG, for example. Okay, so you need to add all of these information to get the gene prediction. Okay, because the ab initio is basically you don't have any reference. You don't have, like, you get new genomes, and then you don't have any like the unknown genome that you need to predict. So you need to use the ab initio. And when you want to study, for example, the one that common like human, or you have you want to study about the E. coli, for example, that is a very common organisms. You can have the reference. So when you have a reference, you need to use this method, evidence-based. So evidence-based, or in a short, we call it extrinsic, which means that it rely on the other DNA sequence. Okay, because it can predict the genes based on the reference. Okay, this is the reference. Okay, so it will predict, okay, based on the all of this reference, the gene itself. So the evidence base is actually, you can have like homologs, okay, homologs from your sequence or your genomes against the genes in the database. So by using this evidence base, most of the genes that you predict can have a like strongly support by the sequence, the reference sequence. Okay, or in the third you can have a multiple program to predict the genes. So basically, maybe you can have like 1, 2, or 3 methods, okay, for the gene predictions, and you can have like a consensus whether okay, these genes predict like this, okay, the second doesn't predict but the third has predict. So you can accept this one because 2 out of the 3 program have agreed that this is actually a gene. Okay, so this is how we can use a third method like a consensus. Okay. Okay, this is all the gene prediction tools that are available. Okay. This is the genes prediction for the non-coding, okay, or the repeated itself, okay. And this is another tools for the gene prediction tools, and for the genome prediction package, okay, and how you can annotate the genome. So basically, when you want to do the gene annotation or you want to assign a function to the genes, the basic tools that you can use is BLAST, okay, basic local alignment, okay, where you can infer the function. I think you guys have been familiar with this website, right? The BLAST software. Okay. Are you guys following? Okay, so this is the program that you can get from the BLAST program. Okay, BLAST N, BLAST P, BLAST X, BLAST T, BLAST N. Okay. All of this program is actually depends on the input that you have and what the output that you want to have. Okay, and the more complicated the process is, the more computational power it needed. Okay, for example, if you use a BLAST N, is N here is referred to the nucleotide, so DNA, and you get the output as DNA. P here is for protein. So if you have protein, you will get the output as a protein. X here is you mean a translate. So you have a DNA, but you need to translate it, and then it can mesh into a one protein. Okay. So if one protein, T plus N, you need to translate the N here, the database. So you need to translate the database. So the computer power could be more than 10%. Okay. If all the DNA that you need to translate into all the DNA in the database, so it's required a lot of the computational power. Okay, so the BLAST result is basically you need to have the cutoff will be less than 0.5 for the e-value. Okay, and then you can have a higher percent of the identity and also the score. So this is how we do the annotation to the genes, okay? For the non-genes, okay, so you can refer to the RefSeq or the Interpro database. For the putative, you can also refer to the RefSeq or the Interpro scan, okay? Hypothetical is basically you have the homolog, but the homolog in the database also doesn't have a function. For uncharacterized is basically have support the transcriptomes that have the gene itself have been mapped to the transcriptome data. Okay, so we know that the gene has been present, but we doesn't know the function. And lastly is novel, which means that it being predicted by multiple programs and also the transcriptome data. So we can suggest that this gene is actually a dubious or novel. I think. Okay, so this is all the other annotation software that you can use, like how we can assign the domain. Okay, the domain is this one. Domain will refer to the function because each of the domain must have a function. So when you use this database, okay, this database is basically the one that can predict the domain. Okay. Enzyme is basically if the gene itself have function as an enzyme, you can get the information in the CAT pathway. And lastly is a BLAST2Go. BLAST2Go, you will have 3 classifications, okay? Molecular function, biological process, okay, biological process, and also the cellular compartment. So this is the format for the GFF, okay. You will have the scaffolds of the gene ID, okay, what software that we did for the gene predictions, okay, the source, the type is added genes, mRNA, exons, okay, a start and stop codon, start and stop of the genes, okay. This one we refer to the score, the phase, okay. The plus minus here is actually it's referred to the forward and also reverse, okay. And then phase, and lastly is the attribute. Attribute is basically contain the name of the genes and all the information that you want to add, okay, whether this gene is actually enzyme or not, you can add all the information in here in the last column. Okay, I think that's all.
Okay, Doctor. Doctor,
Oh,
it's
do
too
you
much
have any
this
question?
too much lectures.
Yes.
It's very old, too much lectures. You can focus the important.
The important is this one.
Where?
This one.
Mhm.
That's why you need to understand how we can get the features, or how we can identify or what information that you can get from the sequencings. Okay, so you will have the non-coding RNA, the genes, the intergenics, the repeats. Okay, and you need to understand each of these component. Okay, what is the genes? What is the small RF? Okay, what is the repeats? All of this you need to understand.
Mhm.
Okay, so basically the genes, what is the gene? Genes is actually consists of the DNA as a basic unit, and genes can also translate into a proteins, and genes is actually unique features for each organism. Oops. So you need to understand all of these features or information that you can extract from the sequencing. Okay.
Mhm.
Try to focusing, okay. You can have, several you need to understand each of these component, okay. And the second part that you need to be focused is this one, gene prediction methods, okay. So you need to understand what is the ab initio, what is the evidence, and what is the consensus. Okay, so you need to be able to that you can elaborate what is the difference between all of these gene prediction method. Okay,
Okay.
yeah, I think that's all.
Thank you,
Any
Doctor.
more
Thank
question?
you.
No. Because this is actually basically the one that's this previous lecture. So we just do some revisions on it. So hope you can have some idea, okay, what is the thing that you need to be focused on. And then I think that's all if you don't have any questions.
Okay. Thank you, Doctor.
Yeah, all the best. Go, is
Okay.
it okay? You're so quiet today. Yeah, I think that's all. Dr. Nick, so you have any more to say?
Yeah, thank you, doctor.
Go. You have any question?
No, thank you.
No. Okay. So hopefully you get an idea, okay, what is the gene predictions and gene annotation is all about, okay, and try to focus part of the one that I have mentioned regarding to the genes predictions and the features that you can get from the genome sequencing and so on.
Doctor, you can share the lectures in email or WhatsApp.
Yeah, you have my WhatsApp.
I think, yeah.
Can you please WhatsApp me first so I can
Okay, you
send
need
it?
research. I think not fine now. I need research.
Because my WhatsApp has been, you know, reset, so all the information is gone. So only the one in the contact that have
Already I'm WhatsApp you.
yeah, okay, thank you. I will WhatsApp you the slide later on.
Wait.
And then you can forward to the girl, eh?
I not find WhatsApp for you.
Where is it?
Okay, I take my WhatsApp in chart now.
Yeah. Where is it?
That's my WhatsApp.
You did WhatsApp me already?
No, my WhatsApp in chart.
Oh, you put your numbers in the chat. Okay.
Yeah.
What is the extension there?
Yes.
Maybe you can add mine.
Okay. Wait.
Okay, and then I will forward the note. We cannot forward here, right?
Okay, I'm WhatsApp you now.
Yeah. You WhatsApp me already?
Wait. I need to search for you. Why I don't find? Wait, Victor, my internet is not good.
Okay. Once you add my number, you WhatsApp me later on. I can
Okay.
I can forward the slide to you.
Okay.
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 Thank you very much and all the best. Okay. I really
Okay.
Okay, all the best. I
Yeah,
will email my slide to you.
Yes.
So if you did not receive my slide, you can maybe WhatsApp me lah. Right?
Okay.
Okay, good luck
Okay.
everyone.
Good luck.
Thank you, Victor.
Bye.
Thank
Well,
you.
thank you, doctor.
Okay. All the best. Okay. Bye.
Bye. Assalamualaikum.
Assalamu alaikum warahmatullah.
Salam.